Here we are on the thylakoid membrane of a cell. On the outside we have the stroma, and on the inside we have the thylakoid lumen. Now, let's take a look at the light-dependent reactions of photosynthesis. First, energy in the form of light reaches the pigment molecules studied in photosystem 2, depicted here in pink. The little pink circles are pigment molecules. This energy excites an electron in a single pigment molecule and is then transferred from one pigment molecule to the next until it reaches the pair of alpha chlorophyll molecules in the reaction center complex. The alpha chlorophyll molecules are located in the P680 reaction center complex, which is named for its optimal absorption of light with a wavelength of 680 nanometers. Once the alpha chlorophyll molecule's electrons are excited, the electrons are physically transferred to the primary electron acceptor, represented here as the shaded pink box. The excited electrons then leave the primary electron acceptor and pass through the first electron transport system. The electrons that go to the primary electron acceptor are later replaced, harvested from a water molecule that is broken apart in the thylakoid lumen through a process called photolysis. Photolysis is the breakdown of a water molecule into 2 electrons, 2 protons, and an oxygen atom. This provides electrons to replace those that left the primary electron acceptor, and provides protons in the thylakoid lumen to create a concentration gradient of hydrogen ions across the thylakoid membrane. Additionally, the single oxygen atom is unstable by itself, so it bonds with another oxygen atom to form an oxygen molecule, which is released into the air as a waste product. Now, let's return to the electron transport system. The electrons travel through the electron transport system on the membrane via electron carriers. While these electrons are being transferred, they are slowly losing their energy, known as an exergonic flow of electrons. The energy given off is vital for the synthesis of ATP. Additionally, while the electrons are passing through the electron transport system, hydrogen ions are pumped into the thylakoid lumen, which further increases the concentration gradient that is used in chemiosmosis. We arrive next at photosystem 1. Here, a process similar to that of photosystem 2 occurs. Light energy is passed from pigment molecule to pigment molecule until it reaches the alpha chlorophyll molecules in the P700 reaction center complex of photosystem 1. Using this energy, the alpha chlorophyll molecules pass electrons to the primary electron acceptor. When electrons go to the primary electron acceptor, replacement electrons passed from photosystem 2 fill this hole in the P700 reaction center complex. Remember, this is different than photosystem 2, where electrons from water fill the hole in its reaction center complex. Now, let's take a look at the second electron transport system, beginning at photosystem 1 and ending at the protein ferredoxin. NADP+ reductase, located next to ferredoxin, catalyzes the transfer of electrons from ferredoxin to the molecule NADP+, which reduces the molecule with 2 electrons and a hydrogen ion. This further increases the concentration gradient on the thylakoid membrane by removing a hydrogen ion from the stroma and creating NADPH, which carries hydrogen to the Calvin cycle for the creation of sugar. So in review, the first electron transport system results in the production of ATP, while the second results in the production of NADPH. The last step of the light reaction is chemiosmosis. Remember that throughout the electron transport system, protons have been pumped from the stroma into the thylakoid lumen. Now, as protons diffuse down the concentration gradient of the membrane from the thylakoid lumen to the stroma via the ATP synthase channel, ATP is formed, which in turn is used to power the Calvin cycle and the light-independent reactions of photosynthesis.